Hello everyone, my name is Youjin Lee from South Korea. It is my honor to have this opportunity to present my work at the Gender Summit 19. The title of my presentation is Gender Factors in Addiction Studies. In this presentation, I will talk about various gender factors considered in scientific addiction research. This work is performed by Gender Innovation Biotechnology Research Group at Ewha Women's University and G-STAR. Recently, the gender issues is becoming crucial both in human and animal studies. As you can see this graph, the number of articles including gender factors in addiction is repeatedly increasing. This tendency may be because the researchers have found clear gender differences in addictive behaviors. Moreover, several major journals started to require the consideration of relevant gender factors from the experimental design. In this study, we tried to identify the gender factors related to addiction in human and animal studies. In animal studies, several artificial, pharmacological, and genetic manipulations are possible. In human studies, it is much easier to investigate the environmental and sociocultural effects. The results from animal studies can be translated to human study, and the new hypotheses found in human studies can be verified by animal studies. Therefore, we will compare difference in gender factors between human and animal studies, and gender factors can be newly adopted to the human and animal studies in addiction. PubMed search engine was used to search all relevant studies about gender differences in addiction. The original scientific articles, including keywords gender and addiction, was chosen, and the papers published in last 10 years for human studies and 5 years for animal studies were selected. Data from selected papers were extracted into summary table. These tables show gender factors considered in human and animal studies. The types of addiction of them also organized into another table. To identify the tendency of which country conducts studies considering gender, we counted the number of studies and classified by nation. United States have the highest percentage of it, followed by Europe. Asia accounted for only 1%. The graph at the bottom shows gender factors considered in human and animal. In cases of human studies, various gender factors were considered, such as an income level, race, education level, family composition, and so on. For animal studies, biological factors distinguishing sex rather than gender were mostly considered, such as hormonal, genetic, and neurobiological factors. There was also difference in the target of addictions between human and animal studies. Not only drug addiction, but also game, internet, and smartphone addiction have been studied in human. On the other hand, animal studies focused on drug addiction. In drug addiction, a variety of drugs were covered by animal studies, whereas human studies were limited to assess better drugs because of legal issues. It is well known that addiction is highly affected by gender factors such as biological sex as well as social factors. For now, it seems quite impossible to implement the experimental setup for animals mimicking human social cultural environments. However, recently, complicated animal behavior experiments are becoming available to study the effect of social networking, even in rodents. Therefore, by developing novel experimental designs, animal study can cover various gender factors. This complementary interaction between human and animal studies will be helpful to figure out addiction problems related to gender factors. Our research group are now trying to analyze the gender differences in animals' alcohol addiction using meta-analysis. Meta-analysis will be the important next step to understand the mechanisms that promote sex differences in alcohol addiction. Moreover, we will keep attempting to analyze the previous addiction research considering gender differences to find out which gender factors have been included or excluded in the humans or animal studies. Thank you for your attention.